Today, I would like to discuss with you the protocol that we use for preparation of cellular lamellas by a cryo-FIB for and transfer in between those instruments. And all of these increase the risk of sample damage and also decreases the throughput of the technology. We first need to culture the cells either on the TM grid, or we need to apply it to the TM grid, then vitrify the sample in a similar ways how we vitrify the samples for single particle analysis, for instance, by plunge freezing. Then we need to transfer that grid into the FIB microscope for preparation of the lamella, and after that, when the lamella is ready, we can transfer again into the transmission electron microscope for cryo-electron tomography. So you can see that from the start to the end, to the data, it can take up to 48 hours, which means that the throughput is still very fairly low, and the goals or the discussion which will follow now will try to focus on how to improve the quality and throughput of the whole workflow. So the test sample which we'll be discussing here is the Saccharomyces cerevisiae, the yeast, which are small cells, roughly 2 to 5 micrometers in diameter, that are heavily used in the molecular biology or cellular biology research as a model system for eukaryotes. We normally culture the yeast in a flask and harvest them in the middle of the exponential phase. And after that, we either concentrate or dilute the sample for vitrification. One important factor also is that one needs to take care is the aging of the culture, because that after, for instance, after 10 to 20 weeks, we see that the cells grow very slow in comparison to the fresh culture. Therefore, it is important to keep the culture fresh and minimize the time that is needed to culture the sample before the vitrification, in order to speed up the whole workflow. So then, once the culture is drawn, we need to vitrify the sample for the cryo for the subsequent FIB SEM. In the case of the suspension cell culture, the cells are not cultured on the grid, but are applied to TM grid and then blotted away and vitrified into the liquid ethane. The major difference in comparison to the standard samples used for single particle here is that the vitrification has to be done with a non-absorbic material applied from the sample side. This is normally used materials are PTFE, so the Teflon, or we have came up with a solution that uses 3D printed pads that you can print from flex material that is standardly available for the 3D printers. Then on the back side, on the other blotting pad, we have a filter paper that then wicks away most of the liquid. And thanks to the blotting with this non-absorbic material, we have the cells still retained on the grid. For the next discussion and next application, we use two different concentrations. We either dilute the sample to OD of roughly 0.5 to 1, or OD 30 to 60, and by that we get two different morphologically different types of specimen in the end, which I will show now. So when we use the lower OD, we end up with small islets, small clusters of few yeast cells gathering randomly on the TEM grid. If we use the high concentration of the or a high OD, if we concentrate the yeast to OD around 30, then we get a monolayer on the TEM of the cells on the TEM grid. And in the ideal case, we also see the grid bars in between the individual squares. Here is an example of what happens if you blot against the filter paper. So if you do use the absorbic material from the front side, most of the cells, or pretty much all of the cells, are removed by the filter paper, and then you end up with the, or one ends up with the empty grid. This is the major difference against the adherent cell culture, like large mammalian cells, which are adhered to the TEM grid and can be blotted against the filter paper because they're retained on the grid surface. One other issue that can happen during the vitrification and transfer to the TEM grid is also the devitrification of the sample, as shown here. So if there is an improper heat transfer between the sample and the sample holder, the sample will gradually warm up, and your cells will devitrify, and you will start to see these shapes of completely dehydrated cells, which we call chips in our lab. So after that, we need to transfer the sample into the so-called FIB-SEM microscope. So what it is, a FIB-SEM microscope? FIB-SEM microscope, or sometimes also called small dual beam microscope, is an electron microscope that has a scanning electron microscopy column for scanning electron microscopy imaging, and besides that, it has a so-called ion column, which in most cases, the ion column is focused beam of ions that are targeted or that are used to image the sample. And in most of the cases, these are gallium ions. Besides imaging, the gallium can, due to its heavy weight, can be used to ablate the material and to remove the not only the biological material, but is used to generate lamellas, of course, also from material science samples. In our case, we use the gallium to ablate the cellular material and generate a thin cellular lamella, which is then electron transparent for the transmission electron microscopy. So how does the such a lamella preparation workflow look like? Here we show schematically the whole process. So first, we screen the grid for the quality, so for surface contamination and cellular content using the electrons, so using the scanning electron microscope, and find ideally cells which are located in the center of the grid squares, such as these two. Then we'll first image the cells in the ions using the gallium ions at very low current, not to damage the cells too much, and find the area of interest, so the area where we want to cut the lamella through. After that, we select the region above that area of interest and below, and use high current gallium scanning in order to remove the biological material. Finally, we carry on in this protocol and thin down the lamella to 100 to 200 nanometers. And such a lamella can be then transferred to the transmission electron microscope for cryo-electron tomography. So here is the whole workflow in the descriptive view. So the first part of the workflow is the inspection of the grid quality, the distribution of the sample, and the surface contamination. If the surface is heavily contaminated, for instance, by ice crystals, one needs to consider whether it is worth to carry on, because the additional crystalline may cause an uneven milling, which can damage the, or which can decrease the, help to decrease the quality of the sample in the end. In order to evenly remove the cellular material, we first use the organic platinum, which is in the gas injection system, the GIS. So we add the GIS protective layer, and after that we so-called cure this layer with anorganic metal. In our case, we primarily use iridium, but normally platinum sputtering is used, which increases the conductivity and makes the GIS layer stiffer. Then we initiate the so-called rough milling, where we use the large currents of the gallium and mill away the initial lamella, while keeping the space in between the two milling regions above and below the final region of interest fairly large, roughly 2 microns. And then we gradually decrease the thickness, decrease the current, and also decrease the width of the lamella in a stepwise manner, and generate a lamella which is roughly 300 nanometers. The last step, the so-called polishing step, is done at very low currents, so between 10 and 50 picoamps. And in our lab, we primarily do it only from one side of the sample to finish the lamella to the final thickness of 100 to 200 nanometers. So as you can see, specifically this part of the lamella milling provides an option for the automation, and that is what we are actually now interested in, and we're developing our own software for the lamella milling, which ideally will be available and soon, and will increase the throughput of the lamella preparation, and also allow to prepare the cellular lamellae in 24/7 fashion, and thus increase the throughput of the whole workflow. So in ideal case, the final lamella has an unperturbed front edge, which is made of the GIS layer, and the lamella appears dark when imaging when imaged with 200 or 2 kilovolt or 5 kilovolt electrons in by scanning electron microscope. When it appears dark like this, not bright like here, that means that the electrons can already penetrate through the lamella, which means that indicates that the lamella is already thin enough to be electron transparent. However, if one of the treatments we performed before is not optimal, we may see different artifacts. So, for instance, we can see if the GIS layer is not thick enough, we may start to lose the front edge of the lamella, see the breakage, and the whole lamella may, after that, crack, losing completely the sample. Or one other option is that we start to see the so-called curtaining artifacts, these stripes, which come from the uneven milling of the lamella, which may be caused by either, again, improper curing of the GIS layer, or maybe the surface contamination, which adds material of different density, and which causes then uneven milling of the lamella. Lamella from these, as these lamellas are fairly, as these clusters are fairly small, and the removal of the material is fast. And we get a good reproducibility when preparing these type of lamellas. On the other hand, the distribution of these clusters or islets on the grid surface is irregular, and we have only very few cells or regions of interest per single lamella. On the other hand, when we use the continuous film, continuous monolayer of cells, the preparation of one lamella is much slower because we need to remove more of the material. We also observed quite frequently cracks in the lamellas, and also we observe the crystalline buffer, so that the buffer is improperly vitrified in between the individual cells. On the other hand, the distribution over the grid surface is very regular, and we have large number of cells per single lamella, allowing to collect large number of cryo-ET data from a single lamella. Also, thanks to the quite uniform distribution of suitable areas for milling, this sample is also very useful for the automation. So in order to remove the bending and cracking of the lamellas, we came up with an additional step, which cuts additional two slices with two areas alongside the lamella direction with the FIB at the initial state. So when we rough mill the first two parts at a high current, before that we first start by milling these two additional cuts alongside the lamella direction, and we've seen that this removes the tension from the lamella afterwards and helps to diminish the cracking of the lamellae. As for the improper vitrification of the buffer in between the cells, which is detrimental then for primarily for the cryo-electron tomography, because it can misguide the focusing and tracking. We found out that addition of 5% glycerol to the buffer or medium just before the vitrification effectively as a cryoprotectant in the buffer and diminishes or completely removes the presence of reflections from a crystalline buffer in the final lamella. As an interesting thing, I want to also point your attention to the fact that we can use the SEM imaging or the image our sample directly in SEM to learn some information about it. This is a SEM image of the yeast cell taken by our already 10-year-old scanning electron microscope Versa, and you can directly see that we can nicely observe the ultrastructure characters, ultrastructure of the yeast cell. So one can expect that with the new microscope with highly sensitive detectors, which is also the case for the SEMs nowadays, one can get much nicer and higher contrast images of the cells already in a scanning electron microscope, and use that, for instance, for ultrastructure studies or as a guidance for targeting the lamella preparation at some point. One word of caution here is that, and this is the same part, this is the image from the transmission electron microscope taken afterwards. The scanning electron microscope and scanning electron microscopy imaging causes a radiation damage to the sample similar to the transmission electron microscopy imaging, and maybe even worse. And this area shows how we damage the sample by scanning over the grid surface or over the sample surface at, in this case, higher current and high dwell times, which means that in order to have such a that this region of is then not suitable for the cryo-electron tomography data collection anymore, and what one even needs to further mill this away, or image the surface at lower currents or lower currents, or at for lower shorter period of time. So afterwards, we have the lamella ready. We transfer it to the transmission electron microscope. The important point here is that the lamella is when transferred to the transmission electron microscope, the lamella is positioned in a way that its direction is perpendicular to the tilt axis of the microscope, and that would mean that we can select the area of interest and have the focusing and tracking area at the same height, and the area of interest is not obstructed by the lamella edges during the tilting experiment. And here we show several videos from the yeast cells. You can observe the nuclear pore here, for instance, the inside of the nucleus, and here a part of the mitochondria, ribosomes inside the cytoplasm. And we also here we also show a couple of images where we have, for instance, can observe a paracrystalline core of the exosomes, and nicely see the microtubules inside the nucleus. Here we again show the nuclear pore, or the details of the mitochondria, including the membrane protein complexes in the mitochondria. All right, so that's pretty much what I wanted to share with you. I would like to thank the colleagues from our facilities, specifically Matthias and Jana, who work on primary work on this topic. Then our colleagues from structural virology group, and our colleagues from Thermo Fisher Scientific Brno, with whom we collaborate on the aspect of the FIB-SEM microscopy. And in the end, I would like to invite you to apply for the access if you're interested for this workflow to our lab. And this is the equipment we have. We're also planning new toys for in the near future, which could further streamline and fasten the whole workflow. And with that, I would like to thank you for your kind attention.